Chapter 1, Section 4, Biologist Tools and Technology. Key Concept. Technology continually changes the way biologists work. Main ideas. Observations include making measurements. Technology contributes to the progress of science. Complex systems are modeled on computers. And the tools of molecular genetics give rise to new biological studies. First, main idea. Observations include making measurements. A wildlife biologist records a description of the alligator mating rituals she observes on her field journal. A pharmaceutical researcher uses probes and computers to measure and calculate the pH of stomach acids. Though very different, these situations are both examples of observation and the use of tools in scientific investigations. Tools serve a variety of purposes. Some tools, such as laboratory glassware and hot plates, allow scientists to set up experiments. Tools, such as microscopes and hand lenses, are used to enhance senses. Rulers, balances, and timing devices enable the gathering of quantitative data. Quantitative data are gathered through measurement, which is the determination of the dimensions of something using a standard unit. The modern metric system, called the International System of Units, or SI for short, is the language for all scientific measurement. The quality of measurements can be described by their accuracy and precision. Precision is the exactness of a measurement. Accuracy and precision are demonstrated by the results of horseshoes tossed in the figure on the screen here. When the horseshoes are close to each other, even if they're not near the goalpost, the results are precise. When the horseshoes are gathered around the goalpost, even if they are not near each other, the results are accurate. When the horseshoes are centered around the goalpost and close to each other, the results are both precise and accurate. Accuracy is the description of how close a measurement is to the true value of a quantity being measured. Second main idea. Technology contributes to the progress of science. Until the late 1600s, no one knew about cells or single-celled organisms. Then the microscope was invented. Scientists suddenly had the ability to study living things at a level they never knew existed. Thus, the microscope was the first in a long line of technologies that changed the study of biology. A microscope provides an enlarged image of an object. Some of the most basic concepts in biology, such as the fact that cells make up all organisms, was not even imaginable before microscopes. The first microscopes magnified objects but did not produce clear images. By the 1800s, most microscopes had combinations of lenses that provided clearer images. Today's light microscopes, like the one shown on the screen here, that you may use, are still based on the same principles. They are used to see living or preserved specimens, and they provide clear images of cells as small as bacteria. Light microscopes clearly magnify specimens up to about 1,500 times their actual size, and samples are also often stained with chemicals to make details stand out. Electron microscopes, first developed in the 1930s, use beams of electrons instead of light to magnify objects. These microscopes can be used to see cells, but they produce much higher magnifications so that they can also see even smaller things. Electron microscopes can clearly magnify specimens as much as 1,000,000 times their actual size. They have even been used to directly study individual protein molecules. However, electron microscopes, unlike light microscopes, cannot be used to study living organisms because the specimens being studied have to be in a vacuum. There are two main types of electron microscopes. A scanning electron microscope, or SEM, scans the surface of a specimen with a beam of electrons. Usually, the specimen's surface is coated with a very thin layer of a metal that deflects the electrons. A computer forms a three-dimensional image from measurements of the deflected electrons. A transmission electron microscope, TEM, transmits electrons through a thin slice of a specimen. The TEM makes a two-dimensional image similar to that of a light microscope, but a TEM has a much higher magnification. SEM and TEM images are artificially colorized with computers so that tiny details, such as leaf pores or stoma, like the one shown on the screen here, are easier to see. Imaging technology is not limited to microscopes. For example, doctors or dentists have probably taken X-ray images of you several times. An X-ray image is formed by X-rays, which pass through soft tissues, such as skin and muscle, but are absorbed by bones and teeth. Thus, X-ray images are very useful for looking at the skeleton, but not so useful for examining soft tissues, such as ligaments, cartilage, or the brain. To image soft tissues, another imaging technology is used, called magnetic resonance imaging, or MRI for short. MRI uses a strong magnetic field to produce a cross-section image of a part of the body. A series of MRI images can be put together to give a complete view of all of the tissues in that area, like the one shown on the screen here. Advances in technology have led to new uses for MRI. For example, a technique called functional MRI, or fMRI, can show which areas of the brain are active while a person is doing a particular task. The first digital electronic computers were invented in the 1940s. They were expensive and so large that one computer filled an entire room. As technology improved, computers, computer software, and hardware, such as probeware, have become invaluable to the practice of biology. Word processing software is used to generate reports. Spreadsheet software is used to quickly and accurately calculate, analyze, and display data in charts, graphs, and other visual representations. The use of probeware in conjunction with computers allows both data collection and analysis. Probeware are measuring tools that can take constant readings of data such as temperature and pH. When probeware is connected to a computer, the data can be calculated and analyzed instantly. Third main idea: complex systems are modeled on computers. Computer-based technology has greatly expanded biological research. As computers have become faster and more powerful, biologists have found ways to use them to model living systems that cannot be studied directly. A computer model simulates the interactions among many different variables to provide scientists with a general idea of how a biological system may work. Computers can model complex systems within organisms. For example, computer models are used to study how medicines might affect the body, as the ones shown on the screen here are doing. Scientists have even used computer models to find out how water molecules travel into and out of cells. The scientists made a computer program that took into account more than 50,000 virtual atoms in a virtual cell. The computer model showed that water molecules must spin around the middle of a channel, or a passage into the cell, to fit through the channel. Water molecules have a specific fit that other molecules could not match. Computer models also help biologists study complex systems on a much larger scale. Epidemiology, which is the study of how diseases spread, depends on computer models. For example, a computer model, like the one shown here, can predict how fast and how far a disease might spread through a herd of cattle. A model can calculate the number of cattle who might get sick and suggest where the disease could spread to humans through eating contaminated meat or other sources. This study cannot be done with real cattle and people. Computer models are used when an actual set of experiments are not safe, ethical, or practical. However, all models have limitations, and they are not able to replicate exactly all aspects of the system they are showing. Fourth main idea: the tools of molecular genetics give rise to new biological studies. Computer-based technologies, such as those shown on the screen here, have led to major changes in biology, but perhaps the greatest leap forward in our knowledge of life has happened in genetics. In just 40 years, we have gone from learning about how the genetic code works to changing genes to implanting genes from one species into another. What is a gene? A gene is a segment of DNA that stores genetic information. Our understanding of the DNA molecule has led to many technologies that were unimaginable a few decades ago. Genetically modified foods, transgenic plants and animals, even replacement of faulty genes. These advances come from molecular genetics. Molecular genetics is the study and manipulation of DNA on a molecular level. Molecular genetics is used to study evolution, ecology, biochemistry, and many other areas of biology. Entirely new areas of biology have arisen from combining molecular genetics with computer technology. For example, computers are used to quickly find DNA sequences. Through the use of computers, the entire DNA sequences or genomes of humans and other organisms have been found. Genomics is the study and comparison of genomes both within and across species. All of the information from genomics is managed by computer databases. By searching computer databases, a process called data mining, a biologist can find patterns, similarities, and differences in biological data. Suppose a biologist identifies a molecule that prevents the growth of cancerous tumors. The biologist could use computer databases to search for similar molecules. This is the cutting edge of biology today. Imagine where biology will be when your children are in high school.